Animal.
animal tissue, right? Isolation of DNA from animal tissue. So either you can take any tissue of the animal, like suppose that if you want to take a goat liver, then you can take it. Here we do this experiment with the help of the chicken liver. Okay, we take a chicken liver as a source from which we isolate the DNA. So, first of all, we have to prepare the reagent for performing this experiment. The first reagent that we required is a SSC buffer. That is your SSC buffer. So we have to prepare this SSC buffer with the help of the two chemical, that is your sodium chloride and sodium citrate. Okay, if we have to prepare this SSC buffer for 800 ml, then we require sodium chloride around 175.3 gram and sodium citrate will be required for 88.3 gram. Now what we have to do, we weigh these two chemicals and dissolve into the 500 ml of the distilled water. When it is completely dissolved, when it is completely dissolved, then we add remaining 300 ml of the distilled water into it. Clear? Clear my point?
Yes, ma'am.
If you have to prepare it for 800 ml, then you require 175.3 gram of the sodium chloride and 88.3 gram of the sodium citrate. It will be dissolved into the 500 ml of the distilled water, and then you have to add the remaining 300 ml of distilled water into it. This SSC buffer is what? It is saline sodium citrate buffer. Saline sodium citrate buffer. Here you added this sodium chloride. That's why this sodium citrate buffer become a saline sodium citrate buffer. Clear? Next chemical that we required is a 2 molar of sodium chloride. Okay. So now it is very easy. Suppose that if you have to prepare the sodium chloride for 50 ml, which formula we use?
Molecular weight.
Suppose that we have to prepare it for 50 ml. So simply weight is equal to molecular weight multiplied by molarity multiplied by volume given divided by 1000. Molecular weight of the sodium chloride is 58.44 gram per mole. When we put all these formula, we get this value. Clear?
Yes, ma'am.
Next chemical that we required is chilled ethanol. 70%. Now, if we have to prepare the 70% of the ethanol in 50 ml, then how we prepare it? Simply, we do 70 by 100 and divided by 50. We get what? 35 ml of the ethanol plus 15 ml of distilled water, right? Any confusion?
No, ma'am.
Okay. Then next we have to prepare our 3 molar of sodium acetate. Molecular weight of the sodium acetate is 82.03 gram per mole, and we have to prepare it is again for 50 ml. So we use this simply the formula weight is equal to molecular weight multiplied by molarity multiplied by volume given divided by 1000. When we put all these value into the formula, then we get that value. At last, chemical that we required is a tris EDTA buffer that I already told you how we prepare it in this lecture. Here, extraction buffer, yeah, this by this way. Clear? So
Yes, ma'am.
this is how we prepare all these chemical. Now, what is the role of these chemical in this experiment? The first one is your SSC buffer. So this SSC buffer is required for to remove the blood clot. It is used to remove the blood clot. Next chemical that we required is a sodium chloride. This one. It is used to break the cell wall. First of all, what we have to do, we remove that blood clot. After that, we break the cell wall. Then this sodium acetate, it is used to neutralize the negative charge on the DNA sugar phosphate backbone. This chilled ethanol is required to to separate the DNA from the solvent. To separate the DNA from the solvent because this DNA are not soluble into the chilled ethanol. And EDTA is used for the storage purpose. Composition and the role of the chemical is clear.
Ma'am, but animal cell doesn't have cell wall, so
Not cell wall, sorry. It is used to remove
cell
the cell. Not cell
membrane.
wall, right, right. Cell membrane, right. I just, it is used to remove the cell membrane. Okay. SSC buffer is used to remove the blood clot. Sodium chloride is used to break the cell membrane. When the cell membrane is break down, then the material which are substances present into this cell just come out. Then we use that sodium acetate which neutralizes the negative charges on the DNA sugar phosphate backbone. Then after that we use that chilled ethanol which is used to separate the DNA from that solvent because the DNA are insoluble into the solvent. Then after that we use this stress EDTA buffer which is used for the storage of the DNA, isolated DNA. Clear?
Yes, ma'am.
Okay. Now, come to the procedure. See, first of all, we take a 2 gram of animal tissue. Then we added 10 ml of SSC buffer for grinding it. And this SSC buffer is added to remove that blood clot. Okay. One more thing, when you added this SSC buffer into the, like, suppose that this is your mortal pistol, here animal tissue are present, and here you added that SSC buffer. So you have to add this SSC buffer slowly so that paste will form. Okay, if you add at once all these SSC buffer into the for the grinding purpose, then slurry is formed. If you add all the SSC buffer at once, then slurry is formed. Okay, after that you have to centrifuge it. Centrifuge it at 5000 RPM for 15 minutes. Then you get supernatant and pellet. You have to discard that supernatant and into pellet. Again, you have to add the 10 ml of SSC buffer. For the homogenization. Okay, if you perform this experiment at microcentrifuge, then this volume will be reduces 1 ml for microcentrifuge. Okay, one more thing, before you do a centrifuge, after grinding, when you do a centrifuge, filter it to remove the extra part of the tissue. You have to remove it. Sorry, you have to filter it. Okay. In case of the microcentrifuge, take 1 ml of that SSC buffer and again perform the centrifuge, and you get what? Same. You get supernatant and pellet. Discard that supernatant, and in that pellet you have to add 2.5 ml sodium chloride. After addition of that 2.5 ml of the sodium chloride, incubate it at 37 degrees Celsius for 30 minutes. You have to incubate it at 37 degrees Celsius for 30 minutes, and after that you have to again perform a centrifuge at 5000 RPM for 15 minutes. Then you get what? You get supernatant and pellet. Now transfer this supernatant. Okay, which is known as a extract. Transfer that supernatant means that extract that supernatant, and in that supernatant you have to add 1/10th volume of 3 molar of sodium acetate plus equal volume of chilled ethanol. again you have to perform the centrifuge at 5000 rpm for 15 minutes, and you get what? You get a supernet, palette, discard, again extract that supernet, and into that supernet again add the equal volume of the chilled ethanol. In this step, you are not going to add the 1/10 volume of the 3 molar of the sodium acetate. You are already added here. Okay. So you have what you do. You simply extract it and add equal volume of chilled ethanol. And then you have to keep it at refrigerator at -20 degrees Celsius for 1 hour. After 1 hour, centrifuge it at 5000 rpm for 30 minutes. At 5000 rpm for 30 minutes, then you get what? You get that supernet and palette. Discard that supernet. Palette should be air dry to remove the smell of ethanol. Then you have to add 100 microliter of TE buffer and store at -4 degrees Celsius for further investigation. Clear? Clear?
Yes, ma'am.
Any doubt, anyone?
Ma'am, as
Yes.
compared to plant cell, why there are so many centrifugation means?
For the purification purpose, because when we take an animal tissue, the purification purpose is like when we have to purify the animal tissue, so it is very difficult as compared to the plant tissue. That's why here are too many centrifuge step. Because why we perform the centrifuge? For the separation purpose.
Yes, ma'am.
Okay, so when we have to purify that particular DNA, we have to do centrifuge at many times. That's why here we do that. Clear?
Yes, ma'am.
Anything else? Okay, so I have two options. Once that I'm going to discuss the second topic, then show you the video both of them, or should I show the experiment video of this experiment? Then after that we will continue the next topic. Choice is yours. Anybody?
Ma'am, show the video of this topic.
Okay. Bluetooth mode.
Yes, ma'am.
Hello everyone, welcome to the ITLS Academy. Hello everyone, welcome to the ITLS Academy. Today we are going another
Yes, ma'am.
important experiment or an excited experiment. Today we are going to isolate the DNA from the animal tissue. So here we are having the chicken liver. We are performing today experiment on the chicken liver. This is our chicken liver. For performing this experiment, we required few chemicals like SSC buffer. SSC buffer is sodium saline citrate. Other than this, we required sodium acetate and sodium chloride. So first of all, we talk about the composition of SSC buffer, that is sodium saline citrate. For this, we prepared in 100 ml, and for 100 ml, we need to weigh 21.9 gram of sodium chloride and 11.03 gram of sodium citrate, and dissolve it in 100 ml, and this SSC buffer solution is prepared. Now we prepared this sodium acetate of 3 molar and 2 molar of NaCl. For making this solution, we need to use a formula, that is weight is equals to number of moles into molecular weight into given mass upon 1000. As we already know, the number of moles of sodium acetate is 3 and the number of moles of sodium chloride is 2. Now we can calculate their molecular weight, and given amount is already mentioned, that is 100 ml. We need to prepare it in 100 ml, and after calculating the value, we get the value and dissolve it in 100 ml to get these compositions. So these are the three basic solutions which are required for performing this experiment. Now first of all, what we are going to do, we are going to take 2.5 gram of chicken liver and grind it with the help of mortar and pestle. As we already weigh 2.5 gram of chicken liver, now we are going to we take 10 ml of SSC buffer, but we need to make sure that we need to make a paste. So we are going to add a little amount of SSC buffer and going to grind it very properly. See, if we dissolve the entire amount in a one time, then it will become a slurry, and we want a paste. That's why we need to add drop by drop to make it a paste. So, and do you know why we are going to add SSC buffer? Okay, let me tell you. So we are going to use SSC buffer so that all the blood clots are removed from the chicken liver. So after making a proper paste of chicken liver and adding 10 ml of SSC buffer, now I'm going to filter it. And for the filter, you can use filter paper. So after filtering the paste, now I am going to pour 1 ml of this paste in each of the appin drop with the help of micropipette. After adding 1 ml of this paste in each of the appin drop, we are going to centrifuge at 5000 rpm for 15 minutes. So after performing the centrifugation at 5000 rpm for 15 minutes, as you can see, the pellets are floating over the surface. Now what we are going to do, we need to discard the supernatant very carefully. We need to make sure that while we discarding the supernatant, pellet should not be discarded. Very carefully we need to discard the supernatant. And after discarding the supernatant of each appin drop, we need to add 1 ml of SSC buffer and further performing the centrifugation at the same rate of 5000 rpm for 15 minutes. Now as you can see, now these are the pellets. Now as you can see, now these are the pellets which were floating on the surface. Now what I'm going to do, I'm going to add 1 ml of SSC buffer in each of the append drop and perform centrifugation for 15 minutes at 5000 rpm. Now here I'm going to add SSC buffer. After performing the centrifugation at 5000 rpm for 15 minutes, as you can see, the pellets are floating over the surface of this SSC solution. Now what I'm going to do, I'm going to discard the supernatant, and after discarding the supernatant, we need to add 1 ml of 5 molarity NaCl solution. Again, remember that we need to discard the supernatant very carefully so that the pellet should not be discarded. As you can see here, these are the pellets. Okay, now I'm going to add 1 ml of NaCl solution in each of the append drop, and after adding 1 ml of NaCl in each of the append drop, we need to incubate these append drop at 37 degrees Celsius for 30 minutes, and after incubation we need to centrifuge it again at 5000 rpm for 15 minutes. So here, after adding NaCl solution in each of the append drop, we need to incubate it for at room temperature for 30 minutes. So after performing the centrifugation at 5000 rpm for 15 minutes, now what I am going to do, I'm going to take the supernatant further. See, this is very important. Till now, in all the isolation processes, we can take the pellets forward. But now, when we perform the isolation with the help of animal tissue, we need to take supernatant forward. So now I'm going to take the supernatant into the another append drop, and after taking into it, I am going to add 100 ml of 100 microliter of 3 molar sodium acetate and same 100 microliter of chilled ethanol. So now as you can see in append drop, I already marked it as 100 microliter. Now I'm going to take 100 microliter of sodium acetate and pour it into the append drop, and the same amount of chilled ethanol is added into it. And after adding sodium acetate and chilled ethanol into it, we need to centrifuge it at 5000 rpm for 20 minutes. Now, as you can see, our append drop is ready for the centrifugation. I'm add 100 microliter of sodium acetate and 100 microliter of chilled ethanol into it. So after centrifuging at 5000 rpm for 20 minutes, now what I'm going to do, as you can see, there is no pellets. If pellets were there, we need to discard them. But there is no pellets, so I'm going to add equal volume of 70% chilled ethanol to each of the append drop. And after adding this 100 microliter of 70% chilled ethanol to each of the append drop, we need to refrigerate at -20 degrees Celsius for 1 hour. Now after 1 hour, I am taking out these append drops from the refrigerator, and what we need to do, we need to centrifuge them at 5000 rpm for 30 minutes. So after this, I am going to put this into the centrifuge, and after 30 minutes we are going to saw the results. So after performing the centrifugation at 5000 rpm for 30 minutes, as you can see, these are the pellets we are having. Now what I'm going to do, I'm going to discard the supernatant, and after discarding the supernatant of each append drop very carefully so that the pellet should not be discarded, I'm going to add tris buffer in each of the append drop, and 100 microliter of tris buffer is going to add in each of them. So see, as now 100 microliter of tris EDTA buffer I'm going to add in each of the append drop, and then we refrigerate it further for the further process. So here our DNA isolation of DNA from animal tissue is completed. Hope you understand this.
So this here.
Yes, ma'am. Yes, ma'am.
So the next is pH meter calibration. So for the calibration of the pH meter, we required a 2 decibel solution. First of all, we required a 2 decibel solution, which third decibel is pH 7 and second decibel is pH 4. Okay. So we have prepared these decibels like pH 7 and pH 4. And prepared by either can be prepared by the capsule or sample, which is available in your lab. And the instruction of the preparation of this buffer are written on that bottle. Written on that bottle. Suppose that in our laboratory we have a capsule. The instruction for the preparation of the instruction of the preparation of that pH difference of the capsule are already written on that vessel. Okay. So suppose that if we have to prepare this pH, now what we have to do, we take out one capsule and twist it. Then after that, the material present into that capsule will be dissolved into the 50 ml of distilled water. When it is completely dissolved, then we add remaining 50 ml of distilled water and makeup volume 100 ml. Same with that, if you have to prepare a vapor solution of pH 4, same process will be applied. Here, we how we prepare these vapor are here.
Yes, ma'am.
Now, if you have a tablet, in that case, I think what we have to do, we have to study the instructions which are written on that bottle. So, what we have to do, we take one tablet and simply dissolve into the 50 ml of distilled water. Then we add remaining 50 ml of the distilled water to make a volume 100 ml. Okay? This is how to prepare our pH for the calibration of the pH meter. Now, start the calibration. So that this is our pH meter. This is electrodes which are attached to the pH meter, and this electrodes are always dipped into the KCl solution. This is your electrode and this is your KCl solution. And this electrodes are always dipped into the KCl solution. Otherwise, it will be damaged. It will be damaged. And when you handle this electrode, you have to handle it very carefully because it is made up of the glass and it can be easily break. That's why we have to handle it very carefully. Now, these CS meters have a screen which is known as a digital screen. Then we have a screen map. No, we have a phone map. This map is for a temperature. Then we have a call map. Then we have a flow. And last one is a function. Before calibrating the pH meter, we have to set the temperature. And this temperature can be set fairly to the instruction given into the buffer, that buffer okay. Before calibrating the pH, we have to set the temperature. And it is according to that. We have that water in which it is written that we have to set the temperature 25 degrees Celsius. Now we set it by moving it at clockwise direction. Then we remove these electrodes from the KCl solution. Then work with distilled water and rub with butter paper. Rub with butter paper or cloth. And then dip into the pH 7 buffer. First, we calibrate pH 7 buffer because it is neutral. That's why we do it. Now, we dip this electrode into the pH 7 buffer and move these function keys from standby to pH at anti-clockwise direction. Then here some values are shown which are most probably less than 10. Now we have to move the call button at clockwise direction and we move it till this value reaches to the 7. This reaches to the 7, we stop. And again, we do this step like remove from, at that time, we remove it from that pH 7 buffer. Then we again wash it with the distilled water, then rub with the butter paper or cotton, and then we dip it into the pH 7. Now, at this time, we have to move the close button at clockwise direction and we rotate it till it reaches to the pH 4. When it reaches to the pH 4, we stop it. Now we switch off that function key. Now our pH meter is calibrated. Again, we wash it and take the pH of our sample, media, or anything. This is how we calibrate the pH meter. It is clear. Calibration of the pH meter is clear. Now I'm going to show you the experiment video of the calibration of the pH meter. Just see and let me know if it is clear or not.
Everyone, welcome to the ITLS Academy. Today we are going to discuss the instrument which is known as a pH meter, and also we know that how we calibrate this pH meter. Basically, the pH meter are work on the principle of potentiometer. Okay. So for the calibration of pH meter, we required a pH buffer. So we required a two pH buffer. One is pH 4 and one is for pH 7. Basically, they are what? They are a capsule. See, I am going to show you the capsule and then I will tell you how we prepare it. So what we do, we simply take a 50 ml of distilled water into a beaker and then we twist it, and after twisting it, the substances which are present inside this capsule are dissolved into that 50 ml of distilled water. Once the capsule are completely dissolved into the distilled water, then we make it volume 100 ml with the help of distilled water. Then we get this solution that is for pH 4 and that is for pH 7. Okay. This is a pH meter. This pH meter have four knobs. One knob is for the temperature. One That knob is called a coil, and this coil knob is for a uh, calibration of the pH 7, and this slope is for the pH 4, and this is called a function key, which is moved to and reaches to the pH. Then pH meter also have an electrode. This electrode are always dipped into a KCl solution. Now I'm going to start the calibration of this pH meter. First of all, what we have to do, we have to set the temperature 25. Now I am going to set it 25. Then after that, we remove the electrode and wash it with the distilled water. See, I'm going to wash it with the help of the distilled water. Then with the help of the cotton, we wipe it. After wiping, we dissolve this electrode into the pH 7. Okay. Now after that, what we have to do, we move that function key to the pH. See, when you are moving it in a display, the values are shown. It is here 5.18 or 17. Now we have to move the call button till we get here 7. Now I am going to move it. See, this is 6.8, 10, 11. Now it is 6.45, 50, 6.76. Now it has reaches.
Moving up not at proper direction. The diamond increases for 6.27. When it reaches the sun, I have to check.
7.0 or 7.1. Now we wait it so that it will become stable. See, now it become a stable. Now we uh, move this button from pH to the standby and remove the electrode from the pH 7 buffer. Again, we wash this electrode with the help of the distilled water. Again, we have to wipe it with the cotton. After wiping it, we have to calibrate the pH 4. This pH 4 is for a acidic. Now, when we are putting this electrode inside the pH 4, we are not touch two knobs, that is your temperature knob and a coil knob. Okay. Now I am going to move this function key from standby to the pH, and after that, when you are moving it here, it is in display. The value are there, that is around 5.13. Now wait for the stability. When the stability get, we move the call button. Here it is 4.9493, 91. Now I'm moving the slope button for calibration the pH 4. When you are moving it, the value are decreasing. Now see it is reaches to 4.20, 4.19, 112, 4.07, 05, 04, 03 and 4.00. Now it is stable. Now wait for the stability. When the stability gets, our pH meter are calibrated. Now the value are stable. Now remove the electrode and again wash with the distilled water. And move the function key from pH to a standby. This is how we calibrate our pH meter. Now our pH meter are ready for taking the pH of the.
That's clear. Anybody have any doubt related to both the experiment, that is isolation of the DNA from the animal tissue and isolation, calibration of the pH meter? Any doubt?
No, ma'am. No, ma'am.
So today the topic is over. Next topic that again isolation of DNA from the other sources, maybe it is human blood. And we will discuss the more topics tomorrow, that is your bioinstrumentation. Okay. In this we will discuss about the instrument that we use in our laboratory. Okay. Like when we look at the instrument like your autoclave, laminar, centrifuge, hot-air oven, incubator, muscle furnace, we will discuss about. Clear?
Yes, ma'am.
Okay. Thank you. We will meet tomorrow.